Ladies and gentlemen, it's my honor to be here to give a speech. As you know, peptide is a very important compound for drug discovery. Up to now, over 70 active peptide approved as drug used in the clinics. But find an active peptide is not very easy. For example, active peptide with 10 amino acid. If use a random screen, it's needed to screen 10 trillion compounds. But if use Huikang's strategy, only 9 round 720 peptide sample to be screened. You will find active peptide composed of 10 amino acid. How Huikang's strategy works, I will illustration later. Our kind of physiological and biochemical activity in the body can contribute to the recognition, interaction, and balance between proteins. This balance is broken. It is disease. Rebuilding this balance, the cure disease. There is not every amino acids in protein gives a contribution to between the protein interaction. Only a few pieces of amino acids resid is called epitope peptide. This is important. Screen and discovery this epitope peptide is a key in research and development of drug discovery. There are two kinds of epitope peptide. One is linear epitope peptide. It's a piece of amino acid in the protein, and this can be found through segmental synthesis or protein degradation. The spatial epitope peptide is another kind. It exists in the protein fold, in the protein folding, in the active form, which is very difficult to be found because denatured or decomposition, this spatial epitope peptide decomposed. So how to find this spatial epitope peptide? My recommendation is from China Peptide Library. And bonding energy analysis of epitope peptide for the ligand and receptor interaction is not each amino acid gives the same contribution. Like this, the left is the linear epitope peptide, and the left is the spatial epitope peptide. In fact, like this, the 10 amino acid epitope peptide is dipeptide or tripeptide gives more energy, bonding energy than others. So find this bipeptide or tripeptide we called core epitope peptide. That is important in the drug discovery, research, and development. Natural epitope peptide, it exists in the physiological conditions. But for the drug discovery, we want to more active epitope peptide. And this more active epitope peptide may not exist in the physiological condition. It's an unnatural epitope peptide. Where to find this unnatural epitope peptide is my recommendation in the China peptide library. The current status epitope peptide screen method. There are a lot of peptide library. It's some natural peptide library, for example, snake, bee, scorpion toxins, body fluid, cell secretions, and artificial peptide library, for example, protein degradation products, chemical combinatory peptide library, phage display peptide library. This library contains hundreds of millions or even billions peptide. Screen this peptide is very difficult. If you use the current method, you need separation first and then screen, or screen first, then separation. No matter which way, it's a huge work. And in this situation, some highly active peptide if they are in very low abundance, it is difficult to find. Another situation is for the phage display peptide library or chemical combinatory peptide library. If two epitope peptide, they are very active and their interaction each other, quench each other. For this kind of epitope peptide, cannot be found. So, for the current status, for the current screen method, you need peptide library, separation, activity test, sequence identification, and synthesis verification, and again, activity verification. At least 4 professional scientists are required. So, find an active peptide is very difficult work. Here is Huikang's strategy. Use Huikang's strategy. Find an active peptide composed of 10 amino acids. Only need 9 round screen and 720 peptide samples. How it works? As illustration here. Step 1, you need a screen method, a receptor and a ligand, label ligand. The ligand and the receptor interaction gives a signal. Suppose it's 100. In this system, put 1 dipeptide. If this peptide did not interaction with the receptor, so it did not interference this signal. If the dipeptide is interact with the receptor, the signal will decrease. You will find dipeptide is active. Even sometimes it's not activity is not very strong, but it's active. The totally dipeptide library 400. So you screen 400 times. If you use 96 plate wire plate and macro plate reader, maybe 5 plate, that's enough. You will find one dipeptide, active dipeptide. Use one as a core epitope peptide for the next step. Yes, for this dipeptide, add one amino acid in its C terminal. 20 amino acid, you'll get 20 tripeptide, and in other side, add one, you get another 20 tripeptide. Totally 40 peptide, tripeptide you'll get. Screen this 40 peptide, you will get a tripeptide have more with a higher activity than dipeptide. Again, this time you only need, if you 196 wire plate. For the step 3, using above, you get the tripeptide. Add one amino acid in one side, you get 20, and the other side you get another 20. Totally 40 tetrapeptide you got. Screen this 40 tetrapeptide, you will get an active tetrapeptide with higher activity than tripeptide. Step 4. At this time, add one amino acid. Similar, you will get the pentopeptide 40. Screen this 40, you will get a hexa, and again and again. Totally 9 rounds. You will get a peptide with 10 amino acid. Your screen samples only 720. That's a Huikang's strategy. You may suspect that is the dipeptide have enough activity, or tripeptide have enough activity? Yes, I pick some reference data to support that sometimes dipeptide or tripeptide is active. This is MSH. It's 14 amino acid. The research results show that the core epitope peptide is a tripeptide. There is an anti-hypertensive peptide. Some is 7 amino acids. Yeah, the last is tripeptide or even dipeptide. Is activity. Tripeptide is more activity. Opposite peptide. Danophen is 17 amino acid. Danophen 13, of course, is 13 amino acid. And dimorphin, and all different piece of amino acid. The shortest one is dipeptide, cortophin. Substance P, yes, it's the 4 substance P is 11 amino acid, and P's 1 to 7, and also for tripeptide and dipeptide, its activity is similar with the P's of 7, 1 to 7. And here is some amino acid, different P's of amino acid. Anti-thrombosis. This data, I show you the dipeptide or tripeptide that have the activity. Mostly may not very active enough, but they have activity. So use this dipeptide as a leading. Add one and one makes activity stronger, and the selectivity higher and higher. Totally, the China peptide delivery and Huikang's strategy. A 10 amino acid epitope peptide, if use a random screen, you need a screen 10 trillion compounds. But if use Huikang's strategy, only 720 is enough. Huikang Huikang's strategy, you only need one scientist to establish a screen method. You do not need the peptide synthesis, purification, sequence identification. No need protein technology. So the China Peptide Delivery, Huikang Biotechnology company will invest 50 million Chinese yuan, synthesis 175,000 peptides to form a parallel peptide delivery. It includes dipeptide, tripeptide, and tetrapeptide with precise sequence named China Peptide Delivery. If you have a screen method combined with the China Peptide Delivery and the dipeptide, tripeptide, and tetrapeptide, you just order from the library. But if you want more longer sequence for pentapeptide and hexapeptide, etc., you need order. Huikang will provide the order synthesis. This is China Peptide Delivery and Huikang's strategy. The China Peptide Delivery to provide a service in 2023. And the website will open at the same time. Here is a sample like this. Here is Huikang's other research project. One is development of MPS II syndrome treatment and recombinant human lysozyme eye drops, a novel gene recombinant collagen's hemostatic sponge, a novel raw cosmetics material OGP. The overexpression of exogenous HIDS in MSSC were trans-infectious of Lent virus. MPS2 is mucopolysaccharidosis syndrome. It is caused by mutation of IDS gene in X chromosome, resulting in a systematic disease due to the abnormal metabolism and accumulation of mucopolysaccharides. Its syndrome symptoms neutral disease dysplasia, skeleton development defects. The aim of this project to development stable and economical, as well as cross blood-brain barrier treatment target of to MPS. The method is establish the IDS knockout mice, treat with overexpression HIDS of MSCs to detect the treatment effect. The IDS knockout mice is established and approved the Chinese patent. The production vector of IDS overexpression and recombination MSC is also successful and approved the Chinese patent. The construction method of animal model of milk polysaccharidosis type 2 and use this is also approved Australia patent. Here is the result. The over expression IDS compared with the control is IDS produced, and the animal knockout, IDS knockout mice animal is also successful. Another project is development of recombinant human lysozyme eye drops. Dry eye disease is a multifactor ocular surface disease. Its symptoms is dryness, foreign body sensation, pain, and others. This product is using human lysozyme, recombinant human lysozyme eye drops, and other composition that is similar with natural tear composition. The production process approved the patent, and its quality test is also approved patent in China and in Hong Kong, in Australia. Animal model experiment shows its dry eye model, and the medium concentration and the high concentration is very effective for this DED model. Here is another recombinant collagen homeostatic sponge. This collagen homeostatic sponge is approved the production, approved the Chinese patent. This sponge animal experiment shows that this sponge have very good wound healing, and there is hair follicle and sweat gland-like tissues. But the black control and the natural collagen sponge know this. So that means use this sponge, the skin will produce the people's skin, hair follicle and sweat gland-like tissues. So it's much better than the natural collagen sponge. And the development of novel cosmetics material, OGP. OGP is containing 14 highly conserved amino acid sequence. It's 10 to 14 in organism which is enabled to induce synthesis of collagen in mice. And use this as a cosmetic material. Can show the anti-wrinkle activities. Here is experimental, the safe experimental is the different concentration of the OGP 10 to 14 is similar with control. And this here is collagen 1. It's increase collagen 1's production and inhibit MMP3. Here is other Huikang's international project, mainly cooperate with Australia's university. That's all my presentation. Thank you very much. Huikang will provide you with great support.